Hello, my name is Sciuscio, and today I'm going to talk about the toxicological assessment of flavored e-liquid in SPRAGUE DAWLEY rats in an OECD subchronic inhalation study complemented with system toxicology endpoints. Smoke-free products have the potential to replace cigarettes in the near future. An important aspect for the success of these products is the use of flavoring substances that, as shown in previous studies, may help to maximize product acceptance and guarantee full conversion of adult smokers. Although the vast majority of flavoring substances using smoke-free alternatives are generally recognized as safe when using food, very little is known about their effects when used via inhalation. In this work, we assess the toxicity of a flavored e-liquid in a 90-day inhalation study. And for the preparation of the test item, we selected a vehicle which contains nicotine, PG and VG, and a flavor mixture containing 28 different flavoring substances representing different chemical and metabolic classes. In our study, we had 6 different groups of animals. 3 of them were exposed to an increasing concentration of a flavoring mixture, including 28 different flavoring substances. We also had a vehicle control, 2 comparator groups, 1 exposed to an aerosol containing PGVG nicotine, and 1 containing PGVG and flavors. These comparators helped us to gain a better understanding about the possible synergistic additive effects between nicotine and flavoring substances. Moreover, apart the classic endpoints described in the OECD 413 guidelines, we assessed a series of biomarkers of exposure, respiratory physiology, pulmonary inflammation, and finally we investigated the global transcriptomic changes in nasal epithelium, lung, and liver. The test atmosphere characterization data revealed that the aerosol generation was under control since all results, nicotine, PG, VG, fall within 10% of the target concentration. Moreover, as shown in figure D, the L-carbon, quantified as representative of the flavor mixture in the test atmosphere, was found to be within the expected concentration. The correct aerosol uptake was confirmed by measuring different biomarkers of exposure in plasma. As you can see from the graph, the measured concentration of cotinine, a nicotine metabolite, and L-carbon reflect well the concentrations of nicotine and L-carbon measured in the test atmosphere. We also measured the concentration of 2 additional flavoring substances in plasma, linalool and citronellol, further confirming the correct aerosol uptake by the animals. Pulmonary inflammation was assessed by looking at both free lung cells and inflammatory mediators in the bronchoalveolar lavage fluid. The results showed no significant increase in the number of recovered cells and minimal changes in the inflammatory mediators, suggesting the absence of pulmonary inflammation. Hematology revealed minimal adaptive changes in the total white blood cell count. These effects were consistent with our previous findings and were resolved after the recovery period. Clinical chemistry analysis revealed a non-statistically significant trend toward the energy metabolism parameters like cholesterol, glucose, and triglycerides. Again, these effects were reversed after the recovery period. Transcriptome analysis of the respiratory nasal epithelium, lung, and liver revealed minimal differentially expressed genes following exposure to flavoring substances. In contrast, and in agreement with our previous findings, the effect of nicotine on liver transcriptome were evident, with a few hundred genes up and down regulated. Organ weights showed the typical changes associated with the procedure-related stress response, including higher weight for the adrenal gland and lower weights for thymus and spleen, mainly in groups exposed to nicotine-containing aerosols. Nicotine exposure also resulted in a higher liver weight, especially in female rats, consistent with our previous findings. Histopathology of the respiratory organs revealed a low incidence of changes in the test item exposed rats. Only in the larynx, which is the most sensitive site, we observed a minimal to mild squamous cell metaplasia in all groups, although the effects was reversed after the recovery period. The quantitative morphometric analysis highlighted a slight trend toward increase in thickness of the mid-base epiglottis following nicotine exposure, with the majority being non-statistically significant. In conclusion, the results generated confirmed that the aerosol was well generated and taken up by the animals. There were not significant changes in mortalities, gross pathology or ocular findings. The biological effects related to the test item exposure were limited and in line with our previous findings. Analysis of the bronchoalveolar lavage fluid suggests the absence of pulmonary inflammation. Histopathological assessment revealed only subtle adaptive and reversible changes. The flavoring substances did not induce a measurable biological response on the transcriptome level. Finally, no apparent additive or synergistic toxic effects were observed among the substances at the tested level. Thank you so much for your attention.